Hello, my name is Natalia. I'm a PhD student at Professor Rafael Najmanovic's group, and today I will be talking a little bit about the dynamics of viral glycoproteins, especially the spike protein from coronaviruses, and how it impacts evolutionary fitness and how we are able to simulate it. Well, the spike protein has different conformational states, so here you can see the closed state, the open state, where it opens RBDs, receptor binding domains, and it needs to be in this open conformational state in order to bind to the receptors of host cells, like case 2 in case of humans. To do this work, we use ENCOM, which stands for Elastic Network Contact Model, and it is a coarse-grained normal mode analysis method that was previously developed in our group. You can find it here as a Python package. ENCOM has different functions that we use. First of them is the dynamical signature. That is a kind of a calculated B factor of this oscillating structure of residues kind of connected by springs. And it gives us as output a vector with different values associated with the flexibility of each residue. Second function is the vibrational entropy, which is a measure of the full flexibility of this structure. Third is transition probability, which looks at how the eigenvector that describes the movement of each one of those residues is overlapping with the movement needed to go from one conformation to the other. And the conformational ensembles that generates a group of slightly different structures based on the normal modes and the possible mutations, possible motions of those residues. Well, our work started in the first wave in Europe in the summer of 2020, when one specific mutation, D614G, apparently was very successful. So we modeled that structure, that mutation, the structure with the mutation, and compare it to the wild type, compare the dynamical signatures both for the closed and the open state. For the closed state, we saw that this mutation made it more flexible, especially around the mutations, and for the open state, we saw that this mutation made this structure more rigid, especially in the chain that is open in the structure, around the N-terminal domain and receptor binding domain as well. So it made us create the hypothesis that this increase in the flexibility of the closed state would make it more willing to open, and the decreased flexibility of the open state would make it less prone to close. The only other variant that we had at the time to compare it to was the old SARS-CoV, so we compared it to the structures of the old SARS-CoV, expecting to see opposite results, and this is what happened. So the old SARS-CoV was more rigid for the closed structure and more flexible for the open one. We then use the vibrational entropy, which is only one measure instead of the measures for every single residue, to do a high throughput evaluation. So we performed over 17,000 single mutants, every possible single mutant for most of the spike structure. And we saw that even though the 614G that is around here had interesting results, we had many positions and mutations that had much more interesting results, much higher VDSs. So the VDS was a measure that we created to consider both the delta vibrational entropy for the closed and the open conformations. So we took our top candidates as more transmissible candidates with higher VDSs, so making the closed structure more flexible and the open structure more rigid, to do open occupancy estimation. This open occupancy estimation was made coupling this transition probability with a Markov chain, and we optimized two parameters, one for the transition probability and one for the Markov chain, using the 6 experimental points that we had available at the time. At this time, this 614G theory on why it was successful had already been experimentally proven. And we also used the bottom 20 for the VDSs in order to compare the results. And here we have the estimated occupancies. So we actually did have many of those mutants showing a larger open state occupancy than wild type, that is here at the 25%. And for the bottom candidates, we had almost a locked state with open occupancy around 1%. And interestingly, lots of our top candidates, after later appeared in new variants, especially alpha, beta, and gamma. So we were able to predict, for example, the N501Y mutation that had an open state occupancy estimated to over 50% in our evaluations. Everything that I just presented is available in this article for the spike protein. What I'm currently working, though, is a little bit different. I'm trying to evaluate interfaces. Why? Because we saw with the new variants from Delta on that what was being selected was not the open state occupancy anymore. It was probably immunescape. And actually we saw a decrease in the open state occupancy, maybe related to the fact that the receptor binding domain is a very important epitope. So when Omicron appeared, multiple structures were published in record time, and those structures were associated with different lists of interactions and estimated effects in binding strength to the receptor, for example, and those differences were justifiable when we looked at how different those different structures were. So in order to overcome that, we decided to work with the conformational ensembles that ENCAM is able to produce for us. So here we have, for example, one of the preliminary results for the BA1, which is called Omicron, one of the Omicron, the original Omicron. And here we have the positions that had differences in interactions for the receptor binding domain for each one of those four references, for example. And using conformational ensembles, we were able to group all of those interactions that were changing, and we were able to see a significantly different interaction considering all those distributions. That here are 32 slightly different conformations. Well, please come to talk to me if you have any doubts and I can explain it a little bit better. Thank you very much, and yeah, thank you.